Hello my dear students and welcome back to my channel. In today's video, we will perform an activity for testing the presence of proteins in the food sample. This test is also known as Biuret test, and for this activity we will need the food sample, and in food sample I have taken an egg which is the source of protein and also the pulses which are also the sources for protein. For negative sample, I will be using water, and for this activity, the solutions or the chemicals which we will need are copper sulfate solution and sodium hydroxide solution. Just two things we need to perform this activity. So let us start this activity. So in one of the tubes, I will add first of all water, which will serve as the negative control. Then in the other test tube, I have smashed these pulses and I will add these pulses in another test tube. In this, the pulses tube, I will add water to dissolve it. So here I have added water to this sample. Then in the last tube, I will be adding the egg white. Just the white portion of the egg I have taken. Now in these tubes I will add, first of all I have to add sodium hydroxide solution to each of the tube. So I will add 2 drops of sodium hydroxide in each of the tubes. And then I will add copper sulfate solution to all the 3 tubes. So this was our just plain water, and here you can see the color of the copper sulfate only. But in this tube where pulses was kept, here the sample will turn purplish in color. We can see the difference in both the tubes, and in case of egg also we will find that the color of this copper sulfate will turn to purple. What happens in this case is that the sodium hydroxide is added to create an alkaline environment, and in that alkaline environment the copper sulfate will react with the peptide bonds of the protein, and the copper ion will form a bond with the nitrogen of the peptide bond and change its color to purple or mauve color. We can see the difference in the color of both the tubes. In case of, and let me add some more. So we can see now, we can see the color has turned to purple. Let us add NaOH to this also to see what will happen to the color. But here no color change even after adding NaOH, because I think in first case the NaOH was not sufficient. Now we can clearly see the difference between the color of all the three tubes. The first tube is water, which is a negative sample. The second tube is the pulses, and the third tube is the egg white. And this purple color formation indicates the presence of protein in the sample. So this was all about today's video. We'll meet in next videos with more different activities. Thank you all.